Hi, I'm Diana, and I'm very pleased to be here with this talk about the first findings of my PhD. So, plants exhibit a diverse set of traits that don't always vary independently. For that reason, identifying the main independent dimensions of tree variation may allow us to understand the extent to which plant traits are related and also the trade-off behind those associations. For instance, Diaz and collaborators use a very simple principal component analysis to examine the worldwide variation of 6 major plant traits. In this graph, the traits are represented by arrows and the extreme values of the traits by icons. So the most exciting thing in this work is that they identify two main independent dimensions of trait variation in plants worldwide. The first one is known as the size of whole plant and their parts and varies between taller plants with larger diaspores and a small plant with also small diaspores. On the other hand, the second axis is known as the leaf economics spectrum, which balances the cost to produce leaves with cheaply constructed leaves, rich in nitrogen, and with low leaf mass per area against those leaves that are more expensive to produce. After searching for general patterns of plant functional diversity, we can ask about the factors that shape the diversity of traits in plant communities. And I can give you a quick answer that they are both abiotic and biotic factors through processes known as environmental filtering and interaction filtering, respectively. At large spatial scales, these interactions have been widely tested, but at smaller scales where local factors seem to have a key role, we have less knowledge. Here, we investigate plant trait associations with abiotic and biotic factors for an animal dispersed plant community in the amazing tropical mountains of southern Ecuador. What did we do? We measure both abiotic and biotic factors in 9 1-hectare plots located along an elevational gradient. We recorded the abundance of common and selective trees of fleshy fruit plant species in the same plots. And finally, we measured fruit and seed and also leaf traits in 18 and 33 plant species. We also conducted separate principal component analysis to identify the main independent dimensions of trait variation for fruit and seeds and also for leaves. Here you can read these findings. After that, we ran an RLQ analysis and we found that the general association between the main independent dimensions of trait variation identified and the abiotic and biotic factors were larger than expected under random community assembly. Let's look at the details. In this graph of an RLQ analysis for fruit and seed traits, red arrows are the main independent dimensions of trait variation. Blue arrows are abiotic or biotic factors. Closer arrows pointed in the same direction show positive associations, while arrows in opposite directions show negative associations. The main interesting patterns in our system is that fleshy fruited plant species produce few large seeds inside larger fruits in rainy environments, whereas many small seeds inside small fruits were positively related with less rainy habitats. The production of abundant rich protein fruits was positively related with high temperature and at lesser extent with fruit removal. In terms of leaf traits, the production of cheaply constructed leaves was positively related with high temperature, interestingly. And leaf area decreased with increasing rainfall. Our findings suggest that environmental filtering seems to be the main process which shapes the diversity of plant traits in our system. The novel finding here was the similar response of leaf and fleshy fruit traits to temperature and precipitation. I'm convinced that we provide a baseline for predicting changes in plant traits in highly diverse tropical communities in response to global change and associated changes. To end this presentation, I would like to thank all the institutions and people behind this project. Thank you for your attention.